everyone. Thank you so much for coming to my talk. My name is Nicole Provenza, and I'm a postdoctoral fellow in Sheth's lab at Baylor College of Medicine. And today I'll be presenting about our work on long-term ecological assessment of intracranial electrophysiology, synchronized to behavioral markers in obsessive-compulsive disorder. So the overarching goal of my work and this work is to improve the efficacy of deep brain stimulation for obsessive-compulsive disorder, or OCD. So OCD is a psychiatric illness marked by obsessions, which are recurrent distressing thoughts, and compulsions, which are repetitive ritualistic behaviors. It affects 2 to 3% of the U.S. population. First-line treatments include medication and psychotherapy, but they don't work for everyone. 10 to 20% of OCD patients are treatment resistant. And we know that deep brain stimulation targeted to the ventral capsule, ventral striatum, or VCVS region improves OCD symptoms in 50 to 70% of these refractory cases. One of the reasons that DBS might not be 100% effective today is that DBS amplitude is constant, but symptoms aren't. So if stimulation amplitude is set too low, we might still see OCD symptoms, but if stimulation amplitude is set too high, we might see side effects of DBS, including hypomania. So during a DBS programming session, the goal is really to find the amplitude that reduces symptoms without causing side effects. So it's kind of like a sweet spot. But the problem is this sweet spot can change over time depending how symptoms are fluctuating. The current DBS paradigm isn't really set up to handle those fluctuations. Today, DBS is an open loop, where stimulation parameters are chosen during visits to the clinician's office that take place weeks to months apart. So the patient might come into the office for a DBS programming session. DBS is turned on for the first time, and they might say they think they feel better, but then they go home and they experience side effects of VCVS, DBS, or even new OCD symptoms. And weeks to months later, they come back into the clinician's office for another round of DBS optimization, and they don't feel better anymore. So tonic stimulation in this open loop paradigm really fails to address the dynamic nature of OCD symptoms and side effects that may fluctuate over minutes to days in between these clinical visits. So we're proposing that automatic detection of mental states related to symptoms and side effects might improve efficacy of DBS for OCD. What this would look like is we would record neural signals continuously from the brain, use those signals to classify relevant brain states such as an obsessive-compulsive symptom state, and then control stimulation parameters accordingly to relieve symptoms. So there are several key challenges that need to be addressed in order to get here and to realize an adaptive DBS system for OCD. The first two involve better characterizing symptom states. So first, there's a lack of established physical readouts of symptom state in OCD and other psychiatric disorders. Next, there's uncertainty in how OCD symptoms vary over time at home. And the next one involves neural biomarker identification for classification of relevant brain states. So today there's no putative neural biomarker for any psychiatric disorder. And until recently, we didn't have the ability to record intracranial neural data at home. And since home is where these symptoms and side effects are taking place, this is really where we should be identifying our biomarker. So we need the ability to detect neural biomarkers outside of a controlled clinical setting. So we recently published a paper in Nature Medicine that tackles those three challenges from the previous slide, most notably the ability to detect biomarkers outside of a controlled clinical setting. And as you can see from this really long list of authors, this work is part of a collaborative effort, an NIH UH3 effort between Baylor College of Medicine, the University of Pittsburgh, and Brown University, which is my alma mater, where the overarching goal is to develop adaptive deep brain stimulation for OCD. So in this work, I'll talk about our first 5 study participants, who I'll refer to as P1 through P5. The first 2, P1 and P2, were implanted with the Medtronic Activa PC+S, and the latter 3 were implanted with the Summit RC+S. So across these 5 participants, there's some overlap in principal OCD symptoms, but the OCD symptoms really manifest in different ways across each of these individuals. And I want to make note of the fact that all 5 participants are responders to DBS treatment. And the Summit RC plus S is rechargeable and allows for wireless data transmission. And that enables streaming of time domain intracranial electrophysiology, both in the clinic and at home. And this is something that's not possible with the PC plus S system or any other therapeutic DBS platform that's currently available. So in this talk, I'll focus on the data gathered from the 3 participants implanted with the Summit RC plus S, P3, P4, and P5. So during DBS surgery, the leads were placed in either the bed nucleus striatum terminalis, the BNST, or the ventral striatum based on awake intraoperative testing. So here I'm showing P4 as an example, where the tip of the right lead was targeted to the bed nucleus striatum terminalis, which is just posterior to the anterior commissure. And the anterior commissure here is the orange fiber tract highlighted on the slice. And the tip of the left lead was targeted to the ventral striatum, which is just anterior to the anterior commissure. The stimulation and sensing contacts are indicated by the color of each of the 4 spheres here, where the sensing contacts are the green ones that flank the red stimulation contact. And the highest contact on both sides was unused. And I want to acknowledge Raisa Mathura and Noam Paled, who were both instrumental in terms of putting these nice brain figures together. So after surgery, the participants returned bi-weekly to monthly to the clinic for DBS programming adjustments and some other behavioral and electrophysiological testing. So here I'm showing clinical outcomes for P4 on that schedule where the participant returned to the clinic. The DBS surgery and the first DBS programming session are marked by the vertical black lines with DBS on at time 0. And I first want to draw your attention to the Y box, which is the green line, which stands for the Yale-Brown obsessive-compulsive scale. It's the gold standard clinical assessment for diagnosing OCD. So the higher the Y box, the more severe the OCD symptoms. The star marks the point at which the participant became a responder to DBS, according to Y box, at about 30 weeks. Next, I want to draw your attention to the Young's Mania Rating Scale, so the pink line, which is used to assess hypomania as a potential side effect of VCDS, DBS, which you can see is fluctuating over time. The other 2 scales here are the Y box 2, the blue line, which is another measure of OCD symptoms, and the HDRS, a measure of depression symptoms. So we have data through these clinical scales on how the participant is doing when they come into the clinic on about a monthly basis. And these clinical scales typically evaluate symptoms over the course of the previous 2 weeks. But one big question we need to answer in order to find a robust neural biomarker is, how is the participant doing in between clinical visits, and how are the symptoms changing on a daily or a moment-to-moment basis in natural environments? So the what's going on in between visits question is really what we're trying to get at with this work. So to get at that question, we're streaming intracranial LFP both in the clinic and at home, and it's time synced with behaviors. So in the clinic, we're able to estimate changes in affect in response to changing stimulation parameters over short timescales by estimating positive affect via automatic facial affect recognition, or AFR. And at home, we've made an effort to maximize the quality and utility of our recordings by asking participants to record in a really wide range of scenarios. They record just daily, doing whatever they would be doing normally, during exposure and response prevention, or ERP therapy, during sleep, during behavioral tasks, and during multi-day sprint recordings, which are just long recordings that happen over the course of multiple days with breaks for recharging. Participants can control their own recordings at home by using a patient-facing application. which was developed by researchers and software engineers at Brown University, David Chang, Fernando Jelin, and Brett Rohr. And participants can launch behavioral tasks from the playground tab of the recording application. And there are also automatic impedance checks at the start of each recording. So each time the participants do a recording, we ask them to rate their intensity of OCD symptoms using a phone application tailored for OCD. So in this talk, I'll be focusing on the self-reported intensity ratings, which is rated by using the slider shown on the phone from 0 to 10. And the participants are also rating mood, sleep, and food intake. So to give you an idea of what the first programming session might look like, I'll show a video of participant P5. So here we're recording from top to bottom here in the right column, heart rate through ECG and blood volume pulse, 64 channels of EEG, and then we have LFP and acceleration from the RC+S, along with DBS amplitude adjustments. So this video of the participant's face was used to estimate positive affect or positive valence on a 0 to 5 scale based on the intensity of 2 different facial action units related to cheek raising and lip stretching that happens during a smile. So as you watch the video, look for positive affect to increase or decrease with smiles as DBS amplitude changes.
That's weird. Weird. You guys are shaking my body. It's odd. It's weird. My inside's not shaking.
We're back to where we were.
Okay, it's not shaking. How weird. It would literally, when you turned it off, I didn't even realize that it went back to doing this on it. I didn't even know I was doing that.
Yeah.
It's not doing that right now. Oh my gosh. It's like the best thing ever. That's cool. Yeah. Cool. Weird. It's not, like I'm not internally trembling, like fear-wise. Oh, it's different. Cool. That's probably the biggest thing I can feel different, is there's not like a, other than just feeling like a breeze feeling, but other than that, those are the two
Why are
things.
you tearing up?
Because it feels nice that I'm not shaking, and I didn't even know I was shaking. Like, it feels, it's a good thing. Like, I didn't realize it till you turned it off, and it like, my stomach inside felt like it was doing this. And I was like, oh, am I more anxious again? Like, why is that? Wow. Okay. Guys, this is so freaking cool.
Yeah.
Holy crap.
So as we just saw, this was a really emotional and happy moment for this patient. This is the first time she felt relief in a really long time, and you can actually see this emotion getting picked up in the data. So to take a second look at that, we're estimating positive affect intensity in response to changing stimulation parameters. And here we saw a peak in positive affect at smile during the portion of programming when stimulation amplitude was increasing from 0 to 4.5 milliamps in steps of 0.5 milliamps. So in the future, we're really excited to use this data to predict positive affect based on changes in the neural data. So at home, we're monitoring symptoms in between clinical visits and over longer timescales. So these plots show the self-reported intensity of OCD symptoms that the participants are reporting using the Rune application on a scale of 0 to 10. So from top to bottom, we have participants P3, P4, and P5. And the colored points are the symptom intensity ratings. And the solid line shows the Y-box2 score, where higher Y-box2 means more severe OCD. So as I mentioned, all 3 participants are responders, and the time point at which they became responders is indicated by these stars. And so P3 and P5 only really recently reached responder status, but P4, who reached responder status at 30 weeks after DBS on, continues to have pretty wide variation in symptom severity over days. And we find this variation really interesting because it shows that even after responder criteria is met, there are still good days and bad days, and symptoms are still fluctuating. And this point acts as further motivation for developing an adaptive DBS system that can respond to these fluctuations in symptoms and reduce those symptom intensity ratings in the long term. So each time the participants do a recording, again, we ask them to report their intensity of OCD symptoms using that phone application. So we can associate each recording that they do with a particular symptom level. And this bar chart summarizes the amount of data in hours that we've collected across each symptom intensity level, along with how much data we've collected during behavioral tasks, during exposure and response prevention therapy, and in total we've collected over 1,000 hours of LFP across these 3 participants. And notice that the majority of the data has been collected by P4, so this teal color. This participant has been in the study the longest, and most of the data collected by P4 is associated with symptom intensities of below 3 out of 10. So even though this participant reported a wide range of OCD symptoms after responder status, as I showed on the last slide, they're really only doing recordings of neural data when they're feeling well. We generally noticed that this is a trend across participants, which speaks to an underlying reporting bias where participants are more likely to engage with the technology when they're feeling well than when they're feeling bad. So we're also doing multi-day sprint recordings, which are long recordings over multiple days, where ideally the only breaks in recording are for recharging. So here I'm showing an example sprint recording where participant P4 recorded for over 30 consecutive hours with a few breaks, and the participant recorded heart rate and acceleration on the Apple Watch, LFP and acceleration on the RC+S, and they submitted OCD symptom intensity ratings on about an hourly basis, except for during sleep. And the 90 seconds of data that I'm showing here is just an example of each of these data streams. So P4 did one 3-day sprint recording where there were natural exposures to OCD triggers on days 1 and 2. And this recording really stood out to us as a good opportunity for analysis because we don't often see such wide fluctuation in logged OCD symptoms within the same long recording. And our goal is to develop a classifier that distinguishes between low symptoms and high symptoms. So in order to do that, we need to collect a lot of data associated with both states. So LFP availability over the 80 hours here is shown by the orange shading, and self-reported intensity of OCD symptoms is shown on the y-axis. So to analyze the LFP data, I computed spectral power in different frequency bands of interest for the 2-minute segment of data around each symptom intensity rating, each of the points on the plot. And if there is a change in neural activity in the ventral striatum during symptom provocation, we should be able to pick that up in spectral power of LFP. So here I'm showing results from that analysis, spectral power versus symptom intensity for going across delta band, beta band, alpha band, beta, and gamma band. For left in the first row and right in the second row, BCVS. And I found that BCVS spectral power shows correlations with OCD symptom intensity, where the correlation between symptoms and delta band power showed the strongest correlation. And beta, alpha, beta, and gamma band power showed relatively weaker correlations. So another opportunity to record neural data during OCD exposures and variable symptom intensities is during exposure and response prevention, or ERP, therapy. So the exposure in ERP refers to exposing yourself to the thoughts, images, objects, and situations that make you anxious or start your obsession, whereas the response prevention part of ERP refers to making a choice not to do a compulsive behavior once the anxiety or obsessions have been triggered. And all of this is done under the guidance of a therapist. So during the 2-month cognitive behavioral therapy booster phase of our study, each participant does 13 ERP teletherapy sessions. And this chart just shows the data streams available for each one of those sessions over the 2 months for P4. And I boxed out this particular session of interest that I'll show you a video clip from on the next slide. Here, the exposure was a wart, or an image of a wart on a finger. And you can see the participant is at home and the therapist is in the office. And this plot shows heart rate collected over the whole session, and also throughout the session, the participants asked to verbally rate their subjective units of distress, their SUDS rating. And that tracks with the black line. So the video clip I'll show in the next slide corresponds to the gray shaded area on that plot, where the introduction of the exposure happens at the beginning of the clip and is marked by this vertical black line on the plot. So the participant that you'll see in this video has very severe OCD specific to contamination, and the thought of having a wart is really distressing. So the image helps to facilitate thoughts about having a wart.
Recording on the watch as well.
Can you see this okay?
Yeah.
Okay, so right here is where we're talking.
Yeah, I see it.
0 to 10?
.5. Oh gosh. And now I'm looking at my own hands
That's good.
for one of those.
Mm-hmm. So your OCD is concerned that you could have one right now?
Yeah.
Okay. This is really good work. I know that this was a huge trigger.
Yeah, it is.
Even that you're willing to look at this, to purposely look at this, like that's awesome. Let's keep rolling with this. So what are we doing?
Okay.
Why is this wrong and dangerous? And how are we just gonna resist those urges? We're not correcting this.
Yeah. Okay. You can still see me, right?
Yep.
Huh? Yeah. Okay.
Yeah, I can. SUDS?
8.5. Still very strong 8.5. Oh god.
So this participant identified thinking about warts as a rather difficult exposure and actually had to complete a number of contamination-related exposures over many weeks to even build up to this. So this is a really significant session for this participant. So over all the ERP sessions across all 3 participants, we saw a high range of subjective units of stress or SUDS ratings throughout the session. And for this reason, LFT recordings during ERP teletherapy present a really unique opportunity to capture neural data during fluctuations in symptom severity. So as an example, I'll show results from analysis similar to the analysis of the sprint recording I already showed using a session that I've boxed out here for the same participant in P4. So here, the SUDS ratings range from 3 to 7.5 throughout the ERP session, and the vertical black lines show the time point where each exposure began. And as you can see, there is some habituation after each exposure according to the SUDS ratings plotted. So I analyzed LFT data in the same way as before by computing spectral power and frequency bands of interest for each 2-minute segment around each symptom intensity rating. And here you can also see some interesting correlations between SUDS and spectral power across frequency bands. So while delta band power did not show the strongest correlation here of all the bands, we did find that the relationship between delta band power and symptoms was preserved across planned exposures during ERP and natural exposures during the sprint recording in P4. So the recording platform that I've described enables this type of naturalistic analysis, and future work will determine the degree of consistency of these brain behavior relationships across patients and their potential as biomarkers for ADBS applications. So to summarize the significance of this work, For the first time in patients with a psychiatric disorder, we demonstrate the utility of at-home intracranial recordings, which vastly increase the quantity and ecological validity of the recorded data. We captured over 1,000 hours of LFP data at home across different symptom levels, and we demonstrate the ability to measure VCBS spectral power during natural and planned OCD symptom provocations at home. Using that data, we identified delta band power as a candidate biomarker of high intensity of OCD symptoms. And continued opportunities for long-term naturalistic intracranial electrophysiological recordings. We hope this will propel biomarker discovery for OCD and other psychiatric disorders. So next I'll talk about a few emerging challenges that I think the field faces. So we really need to identify robust behavioral biomarkers. The utility of self-report assessments, like intensity of OCD symptoms, are limited as they rely on patient insight into their own psychological state, which is variable. So we really need enhanced passive behavioral state monitoring. We also really need to automate electrophysiological data collection. More than half of LFP data collected for participant P4 is associated with symptom intensity ratings below 2 out of 10. So until LFP recordings can be triggered without participant input, they'll be subject to the participant's motivation, anxiety, and mood. So we need to decrease participant burden and automate data collection. And lastly, we need to harness network connectivity estimates. Obsessive-compulsive behaviors implicate symptomatic networks. So we need to increase the coverage of our approach to a few key areas, including the orbitofrontal cortex or anterior cingulate cortex. By increasing the coverage, our ability to identify a biomarker and design an adaptive DBS system would improve, leading to better outcomes for severe cases of OCD. And so in the next phase of this clinical trial, we're using electrocorticography or ECOG to record from the orbitofrontal cortex in addition to the ventral striatum. So the hypothesis is that OC symptoms result from hyperactive connectivity between the ventral striatum and the OFC. And this hypothesis is supported by both imaging studies and characterization of functional pathways. So we're really excited to see how connectivity may be related to changing OCD symptom states. So in closing, I'd like to acknowledge my PI, Sheth, my thesis advisor in grad school, David Borton, the PI of the UH3 study, Wayne Goodman, and I'd also like to thank our collaborators at the University of Pittsburgh, Jeff Cohn's team, for their computer vision estimates of facial affect. And I want to thank the Open Mind Consortium for their support throughout data collection. And with that, I would love to take any questions. Thank you.